Good afternoon, everyone. Just to get our program started this afternoon, we're very pleased and honored to have with us, besides a distinguished scientist from Harvard, we have Congressman Dennis Moore from the great state of Kansas. Congressman Moore has, from the day he got here, always considered the NIH one of the jewels of federal agencies. I know he's got to run, so I'm going to ask him to go out of order and just make a few remarks, if he can, about the NIH. Dennis?
Thank you very much. And I want to talk beyond the NIH about medical research in general. And I suppose some people up here joke, my colleagues joke with me about they ought to write a book called What's the Matter with Kansas? And in fact, somebody has written such a book. And it really isn't very funny, because what it talks about is what's going on in Kansas right now in terms of medical research and the state school board, which is up for election this year. If we do it right, I'm hopeful we take back the state school board. So this evolution, I think people can believe in God and science at the same time. And I always talk about Senator Orrin Hatch, who has a 100% pro-life voting record, but absolutely understands stem cell research and knows that it has nothing to do with abortion. And I think the messenger, not just the message, but the messenger is important. And I talked to, we've got a wonderful medical research facility in Kansas City, not in my district, right across the state line, called the Stowers Institute, just a jewel. And they do incredible research there, scientists from all around the world. And I talked to Bill Neves, who's the medical director there, and I also talked later to Jim Stowers and said, we should try to bring somebody like Senator Hatch in to talk to the Missouri legislature and the Kansas legislature and help them understand. I think there's a lot of misinformation and misunderstanding about what stem cell research really is. Nobody can guarantee that it's going to cure all these horrible diseases. But if we don't try, we'll never know, and I think that's what we need to do. We owe it to ourselves and future generations to try. Thank you very much.
Well, we welcome you to the caucus this afternoon, and of course we were delighted to hear from Congressman Moore, and I hope we get that message across to the entire Congress. You know that we've had 160 caucuses, and we always welcome any remarks, any topical requests from any of your offices. We've done Alzheimer's, cancer, prostate, breast cancer. So anytime you have a suggestion that you might want to hear about, please tell us. This year, of course, we've had our chairman, Sherry Bollert, Governor Mike Castle, Russ Holt, and Lois Capps, and we've worked hard on doubling the NIH a few years ago, 1998 through 2003, and a lot of people, honestly and fairly, in these difficult financial, fiscal times, think that the NIH has got enough money, enough momentum to carry on. Now, you all know that's not true. We are really shorting the NIH. We're now, instead of fulfilling requests, we used to do 1 out of 3 from the NIH for research. Now we're doing less than 1 out of 5. And we have a whole group of young researchers coming along. And just the other day, Dr. Zerhouni addressed members of the caucus, and in a concise, wonderful way, pointed out in cancer and heart, in Alzheimer's, in asthma, in children's diseases, in drugs, what progress had been made for those 5 remarkable years when we had the doubling of the NIH. I just want to say finally, you know we're flat funded this year at $28.2, $28.3 billion. However, Senator Specter and Senator Harkin, again on the House, on the Senate side, have already put in $200 million more for the NIH. And that's not a lot of money, but at least it's a beginning. And we'd like you to do, and we're not proselytizing during this caucus. We'd like to have you keep in mind, as the Congress converges to a conference, at some time somewhere in the fall, maybe after the elections, there will be a conference on the labor HHS bill, including the NIH. We want you in your offices to be alert at that time to do whatever you can to fund the NIH. Today, we're going to talk about systems biology, a study of science that seeks to pull together all the individual information that we've learned through sequencing of the DNA, and we're going to gain an understanding of biology at a higher level. We're privileged to have with us today a renowned scientist in the field of systems biology, Dr. Erin O'Shea is an investigator at the Howard Hughes Medical Institute. She's a professor of molecular and cellular biology and director of the FAS Center for Systems Biology at Harvard University. She received her undergraduate degree in biochemistry from Smith College and her doctorate degree in chemistry from the Massachusetts Institute of Technology. She carried out postdoctoral research at the University of California, Berkeley, and the University of California, San Francisco. Dr. Shea studies how cells monitor the environment and regulate gene expression, work that has implication for understanding cancer and many other diseases. We're delighted to have her with us today, Dr. Shea.
Okay. Let's start over again. Okay, so I'm really very honored to have this opportunity to talk to you today. I find it very inspirational that so many of you are interested in the research that we're doing in our labs. And I have to say that I feel incredibly fortunate, even in these difficult times, to be part of the research enterprise. And I'm very grateful for your support and also the support of the taxpayers in this country. So today, as Peter said, what I'm going to do is to talk to you about a new field called systems biology, which really holds the promise to inform our basic understanding of living cells and organisms, as well as to improve human health through its impact on drug discovery and on medicine. So systems biology is a large-scale, holistic view of living systems such as cells and organisms that focuses on the structure and dynamics of networks of interacting biological molecules. And as I go on, I will explain, I think, in much more detail what I mean by this phrase. But the major goal of this field, many people believe, is to explain how the interactions of biological components, which have themselves relatively simple properties, give rise to the amazing and complex behavior that all living systems exhibit. Moreover, we hope to learn from these basic science studies to be able to use the knowledge, the principles that we've learned to engineer new biological properties into organisms, and also to generate computational mathematical models that are able to predict the behavior of living cells and organisms under other conditions. For example, under conditions of disease when something goes wrong inside a cell. So this field, systems biology, is highly interdisciplinary, and it really involves a close collaboration between biologists and physical scientists such as engineers and physicists who spend a lot of time focusing traditionally on systems, typically non-living systems. So this field brings together scientists working in many different disciplines. Much of the driving force for the development of this field of systems biology has been the sequencing of the human genome, as well as the genomes of many other organisms. And this knowledge that we have that comes from the genome sequences can be thought of as a sort of parts list for, in our case, a human cell. And this parts list tells us, of course, what genes we have, and therefore what proteins each of our cells in our body can produce. So this process of converting information that's encoded, that's stored in our DNA sequence, that is in each gene, into a protein, is called gene expression. And I'll remind you later on that this process of gene expression proceeds through an intermediate that's called RNA. Okay, and so although the genome sequence represents, I think, for all of us, a milestone in biology, and has really revolutionized the way we go about doing science now, it does little to explain the remarkable properties of living things. For example, how our cells within our body are able to respond to signals from their environment, and how we develop into multicellular organisms with such precision, that is, so few mistakes. So those of us working in this field of systems biology hope to exploit this information that's provided by these genome sequences, and also to exploit new technology, about which I'll tell you a little bit, to be able to understand, to predict, and also to control some of the properties of living systems. So I can better explain what I mean by the parts list, and how this parts list, in our case, the genes, doesn't really tell us how a cell works, by talking about something more familiar to all of you, and that is a car like that shown on this slide. So if I dismantled this car into its component parts and laid them out on the ground and gave you the full list of these parts, even if I told you the function of each of the components of this car, for example, the car has many screws, and the function of a screw is to hold two components together. From this information alone, function and the parts list, it would be very, very hard for you to figure out how the car actually works. And this is because the car is a system of interacting components that has properties that are much more complex than those of its parts. So as is true of the car, the parts of cells, which are genes, the DNA we've been talking about, proteins, which are the workhorses inside all of our cells, and many other biological molecules, they interact with each other in what we refer to as networks. And so what's shown here in this diagram on the right portion of the slide is a representation of the network of interacting genes, DNA, and proteins for a very simple cell, a yeast cell. So these yeast cells, budding yeast, are involved in the production of beer and wine and bread, and it turns out that they're one of the most studied model systems in basic science. And part of the reason they're most studied is because at the level of the single cell, these simple yeast cells are fundamentally very similar to our own. They contain very similar proteins, for example. And in this diagram representing the network, which looks like a rather large mess, what you're looking at is each of the balls represents a gene or a group of genes, and the lines connecting the balls represent these interactions that I'm talking about. And what you can see immediately, even if you don't know anything about what I'm referring to, is that some of these balls, that is, some of the genes, are much more connected than others. So in the middle portion of this diagram, it's a mess. All you see are things laid on top of one another, connected to one another. Other genes, such as some of those out on the periphery here, are only loosely connected to the rest of the network. Okay, so some sets of genes interact with many others, and others interact with only a few. Subsets of this network that I'm talking about carry out functions within the cell. So for example, this blue sphere here contains genes that produce proteins involved in synthesizing proteins within our cells. So the most important point I want to leave you with regarding this diagram is that because the components of cells are organized into networks within our single cells, and because these genes and proteins are connected through physical and chemical reactions, alteration of one component, so a mutation that affects one of these genes in the network, can propagate to affect many, many other parts of the network. And this really represents a different way of looking at disease, a systems biology view of disease that argues that malfunction of one component can propagate through these networks that exist within our cells, networks of interacting, interconnected components, to affect many other properties other than the one that that particular protein participates in. Okay. So what I want to do today is to give you 3 examples of systems biology research that really represent very different approaches being taken in this field. The first system I'll talk about is one where we think we know all of the parts involved. It's relatively simple at face value, and the parts are all involved in one biological process, and scientists have well characterized the activities of these parts. And what you'll see is that by thinking with this more holistic systems level view of the biological process, we can gain insights about the system that we never would obtain from studying the individual pieces one at a time. So the second example I'll talk about is vastly more complicated. It's one where we don't even pretend to know all of the component parts. And as you'll see, I think even in this complex example, these systems approaches can be very, very powerful. Okay, and finally, I'll tell you about a third example where we use the information that's gleaned from systems and also from traditional biological approaches, studying one molecule at a time. And using these approaches in combination, scientists have been able to engineer an organism to have new, desired, and very useful properties, as you'll see. Okay, so the first example I want to talk about is our circadian rhythms. So these are daily oscillations in behavior and metabolism that I think are familiar to all of you. They account for example, our sleep-wake cycles, and also phenomenon like the folding and unfolding of leaves of certain kinds of plants. So circadian rhythms are characterized by 2 properties. The first is that they occur with 24-hour periodicity. So for example, we initiate sleep with a periodicity of once every 24 hours. The second property of circadian rhythms that I know all of you are also familiar with is that the circadian rhythms are what's called free running, and that is they persist in the absence of any environmental cues, that is, any light-dark cycles that exist in the environment. And this really suggests that the circadian rhythms, our sleep-wake cycles, for example, are a consequence of some kind of internal clock, an internal timekeeping mechanism. And I know you all realize this because what happens when you are transplanted from this time zone to one that's distant, say 6 hours separated, you experience jet lag. And this is because your internal clock is out of sync, you might say, with the light-dark cycles that exist in the environment. And to overcome jet lag, what has to happen is that the phase, the synchronicity of your clock with the environment, your clock has to shift to now be in phase with the new time zone, with the light-dark cycles that exist in this time zone. Okay, so many organisms, it turns out, exhibit circadian rhythms, including the very, very simple one shown on this slide, cyanobacteria. So these are bacteria that appear green because they contain pigments that are used for photosynthesis. And this is, of course, the process that green plants carry out to harness energy from sunlight to power all cellular processes. So it turns out that in this organism, cyanobacteria, the expression of genes, this process of decoding the information present in the DNA sequence, is under the control of the circadian clock, this internal timekeeping mechanism. And so what this does for the organism, it is to allow the cells to alternate different forms of metabolism that, because they're chemically incompatible, can't take place at the same time. And so the way the organism solves this problem, it needs these 2 types of metabolism to go on, is to separate them temporally. And so it does photosynthesis during the day, and it does this other metabolic process called nitrogen fixation at night. Okay, and the way that this temporal separation is achieved is through the control of gene expression by the circadian clock that exists within the organism. So remarkably, it turns out that scientists think that the clock within this simple organism may be fundamentally similar to the clock that we have in our own cells and that many other organisms have. And as you'll see, the bacteria, of course, are much easier to do experiments on than we are. And so the hope is that studying how this clock works in these simple cells will help us better understand clocks in more complex organisms. So scientists have devised an extremely clever way to visualize and measure circadian rhythms in cyanobacteria. So the bacteria are, as I said, rather simple and small, and they don't exhibit any changes in behavior akin to our sleep-wake cycles. So scientists have to use a much more clever strategy to visualize circadian rhythms within the organism. And what they do is to use a strategy that monitors the production of a protein. So it's a strategy that develops a molecular marker, if you will, that can then be measured. And so in this case, what was done was to engineer the bacteria to produce a protein whose expression is under the control of this circadian clock. And this is a special protein. The protein can generate light when it's given the appropriate chemicals. And therefore, when the expression of the protein is turned on by the circadian clock, the cells give off light, which can be detected with a very sensitive camera. Okay? And so that's what you're looking at on this slide, an experiment measuring light given off by these bacteria that have been engineered to produce this special protein under the control of the circadian clock. So the top portion of the slide just shows a time series following, looking down a microscope. These are microscopic images in the boxes here, and you're just following these bacteria as a function of time. So the bacteria are very simple. They appear rod-shaped, as do many bacteria, and what you see over time is that the bacteria grow and divide to produce more cells. Now the experiment is shown below, and this is the experiment where the scientists are measuring the light given off by this protein by a chemical reaction that's catalyzed by this protein under the control of the circadian clock. And so the image is pseudo-colored so that when there's no light detected by the camera, the pixels, the spots here, appear blue or black. And when light is given off, they appear a brighter color, either brighter blue or yellow or orange or red, depending on the intensity of the light that's given off. And so what you can see remarkably is that light is given off with 24-hour periodicity, even though, I didn't tell you, these cells are kept in nearly constant light, okay? So the clock, this really is a dramatic demonstration of the clock is free running. It persists even in the presence of a constant environment, in the absence of any light-dark cycles. And so the periodicity here, of course, corresponds to the 24-hour periodicity of circadian rhythms, just like our sleep-wake cycles. Okay, so studies measuring the clock in these single living cyanobacteria have revealed two absolutely remarkable properties of this clock. The first is that the clock is robust. So time is passed on from mother to daughter cell. When the cells divide, the daughter picks up the timekeeping where the mother left off. Additionally, the clock is unaffected by these dramatic changes in metabolism I told you take place under the control of the circadian clock. Second, the clock is precise. It's as good as an old-fashioned mechanical old timekeeping or wristwatch. It can keep time accurately to on the order of months, okay? And this is not, it's obviously not a mechanical device. This is a clock that's made up of proteins inside this cell. And we know what the proteins are, and this is because work from a large number of labs has led used traditional biological approaches to identify the clock components and then characterize their properties. And in this case, the properties are biochemical activities. The three proteins are called Chi-A, Chi-B, and Chi-C, and it turns out one of them is an enzyme, and the other two, they're rather nondescript. Okay, so we know these are the three of the components of the circadian clock within this organism. We know something about the biochemical properties, but this information doesn't tell us how the clock works. So how you generate these oscillations with 24-hour periodicity that persist and that are robust and able to keep time so precisely. So recently, scientists studying the clock made an amazing discovery, and that is that if they mixed these three proteins together in a test tube, in the absence of any other molecules, in the absence of any cells, anything else except ATP, which is the energy source that powers all cells, what they observed when they mixed these three proteins together with the ATP is they observed oscillations in the biochemical activity of one of these three proteins. Okay, and the oscillations depended on all of these components that I'm showing you here. And this is a spectacular finding. These oscillations occur with the same 24-hour periodicity as you measure for the circadian clock inside living cells. And it tells us that the oscillations in the activity of this protein are what drives the circadian rhythms, the control of gene expression within this organism. These four components comprise the core oscillator inside these cells. And this finding now opens up a whole new area of research that will allow us to figure out how these three proteins interact to form this oscillating system. Right now, it's a huge mystery, and it will help us understand why the clock is so robust and can keep time so precisely. So the oscillations are really an example of a system's property. This is one that arises from the interactions of these component parts. It's not a property that comes from only one of these proteins. And so to study the oscillations, you of course have to study the whole system here, which in this case is well defined, these components. And we're very excited about this observation because, as I mentioned previously, the hope is that we'll be able to understand the origin of these oscillations in this simple organism, cyanobacteria, and the hope is it may help us understand how the clock works inside our own cells to control, which is at the heart of how these sleep-wake cycles are controlled. So the system I just told you about is very simple. The number of parts involved in this oscillator is very small, and we know their identity and their properties, and it's a simple bacterial cell. However, many systems that we really care deeply about are much more complicated, and it's frequently very difficult, if not impossible, to know all of the biological components involved in a given process. And so here you might think systems approaches would have been hopeless, but a recent revolution in technology that's, again, catalyzed by the availability of all of these genome sequences has enabled systems approaches to many, many different problems in different organisms. So one new technology that's commonly employed is a technology called microarrays, which allows scientists to measure the expression of all of the genes in an organism simultaneously to get a picture of which genes are turned on and which genes are turned off. A second technology is called proteomics, and this is used to measure large numbers of proteins all at one time. And I told you about a third type of technology, methods for visualizing and analyzing biochemical processes in single living cells, like the protein under the control of the cyanobacterial clock that gives off light. So what I want to do in the next few slides is to explain a bit about how these microarrays work, and to give you an example, I think a spectacular example of why they're changing the way we do research and really starting to have an impact on medicine. Okay, so all of the cells in our body, of course, contain the same set of genes, that is, the same DNA sequence. And what makes the different cells within our body different is what genes are on and what genes are off. So what distinguishes our hair cells from our liver cells is this process of gene expression. The hair cells express a different set of genes than the liver cells. Okay, so when genes are turned on, essentially a copy of the gene is produced that's called a messenger RNA. And this is essentially an intermediate in the process of gene expression, of converting this information in the DNA sequence into a protein. The intermediate messenger RNA is deciphered by a little machine within all of our cells that then deciphers the information and produces a protein corresponding to that specific RNA corresponding to a specific gene. And the proteins, as I said, are the workhorses within all of our cells. So what do microarrays do? What microarrays do is to measure the intermediate. They measure this messenger RNA. And in doing so, they tell us, the presence of the messenger RNA tells us whether the gene is on or not. Okay, so how do they work? So the microarrays can be used to detect differences in which genes are expressed in two different samples. So for example, if we want to identify the differences in gene expression between normal and disease samples, what we first do is to isolate RNA, this intermediate in the process of gene expression, from the two samples. And this, of course, in doing so, we get all of the RNAs corresponding to all of the genes that are turned on in these cells. We then take the RNAs from the two samples and label all of the RNAs in one sample with a red dye, and all of those in the second sample with a green dye. And so what we really want to know is, for each RNA contained within the sample, is there more in the normal sample or in the disease sample? That's what we want to know, and to figure this out, we use microarrays. And the way this works is we take the RNAs that have been labeled with red and green dyes, we mix them together, and then hybridize to an array. And what happens is that each RNA in the mixture it hybridizes only to the spot on the array. So each spot on this array corresponds to DNA from one gene. And the microarray is essentially a glass slide where we literally spot down liquid containing DNA corresponding to one gene. So each of the spots represents one gene. We mix the RNA samples with these dyes together with the entire slide. And each RNA in this mixture binds only to the spot on the array corresponding to the gene that the RNA came from. Okay? And so what we can do is then to determine whether there's more of a specific RNA in the normal or disease sample, we simply measure the red to green ratio, the ratio of these two dyes, for each of the spots on the microarray representing each of the genes within, for example, our cells. If there are equal amounts of a given RNA in the normal and disease samples, the spot will appear yellow because a 1-to-1 ratio of red and green, of course, makes yellow. And alternatively, if there's more of the RNA in the disease sample, the spot will appear, in the normal sample, the spot will appear red, and if there's more in the disease sample, the spot will appear green. So this technology has really revolutionized the way we do research, and you'll be able to see from my example I'm going to give on the next couple of slides, it's really starting to have an impact, and I think in the future will have a significant impact on medicine and human health. So the application of microarrays I want to talk about is one in which scientists use this technology to examine gene expression in breast tumors that were taken from young patients in which the tumor cells had not yet metastasized to lymph nodes. So what the scientists did is to correlate the pattern of gene expression they observed in individual patients with clinical outcome. And the goal was to see if they could identify a set of genes whose pattern of gene expression would then allow them to predict the clinical outcome. That is, to predict the course of the cancer, whether the patient would get metastases or not. So this is the microarray data shown on this slide, examining the expression of many different genes in many different tumors, where each tumor has been taken from a single patient. So each row in this big diagram here represents the gene expression pattern of a tumor taken from a single patient, and each row represents a tumor, and each column represents a single gene. And so then if we read across a particular row here, that row represents what I'm calling the gene expression pattern for a given tumor or for a given patient. So in this diagram, red color means the gene is upregulated in the tumor. That means the gene is more turned on in the tumor cells than in normal ones. And green means the opposite. It means there's less gene expression for that particular gene in the tumor compared to normal. All the boxes where you see gray or black are where the tumor and normal cells have similar expression of a particular gene. Okay, and so you can see immediately that each patient or each tumor has a distinct gene expression profile. That is a distinct pattern of these green and red squares, right? They do not look identical. However, you can see that the gene expression patterns for the different tumors can be grouped based on their similarities. So for example, those above this yellow line, the solid yellow line, all these tumors all have a similar pattern of gene expression. You know, it's green for many of the genes, and then those on the right-hand side here appear red. Similarly, the genes below the yellow line also have some similarity in the pattern of being red then green. It's sort of the opposite, the inverse pattern. So what these scientists then did is to correlate these patterns of gene expression with the clinical outcome. They followed these patients after treatment several years down the line and correlated the patterns of gene expression with clinical outcome. And the spectacular finding that was made is that these two patterns I talked about, green then red, red then green, correlate with clinical outcome. That is, whether the patient will develop metastases or not. So those patients that have the pattern above the yellow line have a good prognosis. Very few of them, indicated by these white bars, develop metastases. But if you are a patient that's unfortunate enough to have the pattern represented by those below the line, these patients have a high probability of developing metastases. And so this is an amazing finding, that this pattern of gene expression can be used to predict with reasonable certainty clinical outcome. So the result is tremendously exciting because it has the potential to impact how patients are treated. This expression profiling I'm talking about outperforms, it turns out, all traditional clinical parameters used to predict the outcome of this disease in this type of presentation of breast cancer. And what the better prediction does, as I'll describe on this slide, is to allow doctors to deliver better treatment. So for example, 80% of the patients that present with this type of breast cancer can be cured by surgery and radiation treatment alone. But the balance of the patients that present with this type of breast cancer, 20%, need additional treatment that's referred to as adjuvant treatment. This is hormone therapy, estrogen modulation, and also chemotherapy. So the goal is you have this mixed patient population and you need to identify the 20%. You need to pick out these patients that need the extra treatment from the mixed set and then give them the extra treatment. And the problem is that the traditional clinical parameters for predicting outcome are so bad that in order to capture the 20% that need the treatment, more than 60% of the patients that don't need treatment get it anyway. And this is a serious problem because the treatment I'm talking about, this hormone modulation and the chemotherapy, has a lot of side effects. It also drives up the cost of health care. So use of expression profiling as a predictor of clinical outcome is able to capture almost all of the 20% of the patients that need the treatment while reducing to 20% the fraction of the patients that are treated unnecessarily. So this is really a significant advance in our ability to predict clinical outcome in this disease. And it's really, again, a consequence of the development of this technology that initiated in basic science research funded by the NIH and is now being applied to human disease. So the final example I want to talk about is one in which scientists use traditional and systems approaches and principles learned from natural systems to engineer organisms to have a desired property. The example I'll talk about is a very recent one in which scientists were able to essentially figure out a better way to make a precursor of this compound shown on this slide that's a highly effective anti-malarial treatment that's effective even against drug-resistant malaria. So this compound shown on the slide is a natural product. It's found in a plant that's indigenous to China. And the difficulty is that it's hard to purify large quantities of this compound from this particular plant. It's expensive. Moreover, it's even harder to synthesize this compound using traditional synthetic chemical approaches. So we need a new way to be able to make the compound, and we need new anti-malarial drugs. And what these scientists did recently was to engineer yeast cells, like I talked about previously, to produce this, actually a precursor to this compound. And the way in which they did this was to make many, many changes in metabolic pathways that are naturally found in this organism. And they also introduced a number of enzymes that aren't even found in the budding yeast cell. And this, through these studies and some modeling studies, they were able to optimize the production of a precursor of this compound that could then be readily converted into the active anti-malarial drug. And this research is incredibly promising, and many others along these lines, in that it may result, in this case, in a cost-effective way to produce a badly needed anti-malarial drug. The incidence, I'm sure many people are aware that the incidence of drug-resistant malaria is so high, we're badly in need of new drugs, and this disease, of course, kills a large number of people, particularly in the developing world. So systems biology is beginning to have an impact on basic science, and we hope that this kind of research will reveal some of the origins of these complex properties exhibited by living things. Properties like the robustness and precision of the circadian oscillations exhibited by cyanobacteria. The principles we learn from studying natural systems will allow us to make better models to understand and predict behavior in biological systems. And I hope I've convinced you that studies in model organisms, organisms like bacteria and yeast, very simple ones, really serve as a test ground for the development of new technology and also new approaches in systems biology that will help us dissect more complex systems like humans and disease states. I've given you two examples of how systems biology is beginning to have practical applications. I described how approaches like microarrays can be used for the diagnosis of disease and also to improve treatment by providing a more personalized approach. Many believe that the greatest promise systems biology holds and its greatest impact will be realized through effects on drug discovery, where here we hope that systems biology will provide us with a better way to identify targets for drug design, targets that are chosen based on knowledge of these networks of interacting proteins and genes I spoke about. And you might be able to envision how you can use this network to identify a drug target. You might be able to select from the network that is involved in a particular process the sort of Achilles heel in the process. That is, the protein which, when perturbed by the drug, would have the largest effect on the process. And that protein, you might imagine, might be the one that participates in the most interactions with others. And this kind of systems biology approach, we hope, will help identify those targets for drug design. This network-based approach, we hope, will also help us better understand the origin of the side effects of drugs, because as I showed you in this sort of network view of disease, changing one component of the network can propagate to affect other components. And in the case of toxicity of drugs, these are side effects. They're undesirable things that, with knowledge of the network, we might be able to prevent them from happening, perhaps by choosing a better target. We also hope that systems biology will, it already is starting to have an impact in this more engineering-oriented form, where we hope that we will be able to engineer organisms to have desirable properties such as the production of drugs, and also something I didn't talk about, but where there's major effort trying to engineer organisms to be involved in the cleanup of toxic waste. So finally, I'd like to acknowledge the labs whose research I really described. What I told you about was not the work of my own lab, and the data I showed came from the labs shown on this slide. So in the case of the circadian oscillator, this was the work of the Leibler lab at Rockefeller, the Kondo lab in Japan. That spectacular story about microarrays and gene expression profiling for breast cancer is the work of Steve Friend's lab at a company, Merck. The biosynthetic production of this anti-malarial compound I talked about at the end is the work of a rather young investigator, Jay Kiesling, at UC Berkeley. He's an engineer. And finally, I should acknowledge a lot of insightful thoughts in commentaries on systems biology that were written by Hiroaki Kitano and also Lee Hood. So I'll stop there, and I'd be delighted to take any questions you might have.
Thank you, Dr. O'Shea. Let me ask you this first question. Cell mitosis, cell division, is there some kind of a clock that tells cells when to divide?
That is a very good question. So there has been a lot of debate over whether the cell division cycle, so in the cell division cycle, cells have to alternate the replication of DNA, that is the duplication of all of our chromosomes, with chromosome segregation, that is the process of mitosis. And there have been many debates previously about whether this process is a clock with some internal timekeeping mechanism like I spoke about in the circadian rhythms, or alternatively, whether this is a process where the second phase, the second step, in this case we'll call it mitosis, is perhaps only completed after it's sort of a more domino-like effect. And people lean towards this latter explanation and do not really believe that that there is some internal clock controlling cell division.
What about, is it called apoptosis when the cells finally die? Is there,
Yes.
is there a clock within every one of us right now telling us when we're going to meet the Big Dipper?
Okay, so in general, what controls apoptosis is a signaling pathway that's involved in, for example, relaying information about damage within cells, about the internal state of the cell. It's a sort of program, the signaling pathway controls a program that then kills the cell from within, if you will. So that process is not under the control of a clock, so that's the answer to that question. But the interesting point you raise is what controls the sort of timing of our ultimate demise, that is the process of aging. And we really do not understand that, and I'd be just, I don't even know if I have a strong opinion about that. There's so little information available. Yeah.
Yes, please. Would you wait a moment for the mic, please? Thank you.
Thank you. I have two questions from the molecular profiling slide. One was, it looks like you've been able to reduce the people that needlessly get adjuvant therapy to, I think, 20%. That's good. But is there work toward getting that number down farther? And my second question is, it seems like it would be wonderful if someone could quantify the savings to the nation, both in productivity and in actual healthcare outlays, of getting this number way down, and is somebody doing that?
Okay, so the first thing I should say that is that this was a pilot study, I think I said, about 120 patients. And so there are now efforts to replicate this on a much larger scale, because the number of patients isn't large enough to use this, to validate this particular approach, although it's very promising. And actually, as a side, I should say that there is a company in Europe now that produces a test based on exactly that finding I talked about, that profiles breast tumors now. That's being used in Europe and the UK as well.
Can you tell us the company?
I think it's called Agendia. Yeah. But your real question was, first, I only remember the second question. I'll answer that first. So the second question is about the health cost savings.
So that we could spend more money on NIH.
Absolutely. I do not know whether anyone's done that calculation. I am certain there are people who would be qualified and able to do it. I don't know myself. Yeah. But the first question.
The first question was just, are you still, is someone
Absolutely. There's tremendous interest in, for many different diseases. I mean, I sort of just picked this one because I thought it was the nicest example, is why I picked it. But sure, people are trying to, in this case, reduce the 20%. Gene expression alone likely will not do it, and it will have to be gene expression combined with other ways of profiling the tumor. And one thing I didn't say, but I mean it's probably evident, is that so the traditional clinical parameters relied on rather gross morphological examination of the tumors, and these new approaches are, of course, very molecular. And so the signatures people are looking for that have real predictive information are these molecular signatures. One approach is gene expression. Another, for example, is proteomics, measuring the proteins directly. And I'm sure in this case people are doing that. And that approach of measuring the proteins directly has been very effective in other cancers. For example, ovarian cancer. There is a test that's being developed where the accuracy, the it is much higher, is actually closer to 100%. It's something that was developed at NIH, actually.
Yes, sir. Thank you.
Quick conceptual question about systems biology. When I go to that MIT website and look at the parts list, it's not just a parts list, it's the standardized parts list. I'm interested in the standard bit. Are all of the components that you're looking at pre-standardized, or is there any variation in them? If there's variation, how do you decide what the standard one is? It seems like that might be an important classification. And a related point maybe, you've given us a fairly genocentric presentation of your project. I'm wondering, and biologists nevertheless tell us that most complex human traits are a combination of genetics and environment. Is there a role for the environment in your networks and models? If not, is that a problem?
Okay, so the first question regarding a standardized parts list, I don't think it's possible to have a standardized parts list. The parts list is more an operational definition. The parts list means, I used it in two contexts. One, parts list as being all of the biological components capable of being produced by, for example, a single cell in our body. There's nothing standard about that, and other than the proteins capable of being produced in my cells are identical to those that are capable of being produced in your cells. But the answer to that kind of parts list question is organism specific, right? The genome dictates what components are capable of being produced, and the parts list is different for the yeast cell. I use parts list in a different context to mean the parts involved in a specific process. So there I was focusing my attention on a specific process rather than wanting to study everything in the cell. I wanted to understand a particular process, circadian rhythms. There are a limited number of proteins that are involved in that process, and therefore I was defining my parts list based on those that are necessary for the process. And in the case example I gave, they turn out to be sufficient to be able to generate the oscillations in a test tube. So there are different forms of the parts list. Okay, so that's about standard. I'm not sure what those people, I think I know what you're referring to at MIT, and that's a sort of, they're trying to standardize a parts list for synthetic biology to generate a sort of set of, like Legos, like Tinker Toys, that they can use to, in bacterial cells primarily, although not exclusively, to generate desired outputs or properties, where the output might be gene expression with a certain characteristic. That's the sort of standardized parts list they're referring to, but it's not a parts list for a natural system. Okay, then the examples I gave, your comment was they were genome-centric. Yes. Why? Because, and not really because I have so much a bias. In fact, as I said, that's not my work. And the oscillator was not genome-centric at all. But I would say just the microarray was genome-centric. There are many sort of incarnations of systems biology that involve things other than genomes. I tried to give you two flavors of that. So one is this genome-centric view, like microarrays, use them to measure everything, and in the example I gave, tell me something about breast cancer and whether we can use it for some useful purpose. That, you're absolutely right, that's like a genome-centric view of systems biology. It's sort of a, there are proponents in this field like Lee Hood who advocate that type of approach amongst others, but he's a big proponent of that. There's also small-scale systems biology that would be typified by that circadian oscillator, which is why I gave you that example. That's an example where we're not measuring like 30,000 things at once. You know, we used regular biology, the field, used regular biology to figure out the parts involved, study the phenomenon in the whole organism, the whole cell, and then use the small parts list to be able to then hone in on the critical parts and study them in a test tube where we can really manipulate things in a way we can never do inside any living organism. So that's sort of small-scale systems biology that is not genome-centric. But so I didn't mean to imply all of systems biology is genome-centric. In fact, I think as science moves forward, it will become less so. It's just that the genome was readily accessible to us with the advent of high-throughput DNA sequencing, and the other cellular components, such as proteins and many other molecules, are much harder to measure in this large-scale, high-throughput format.
Yes, back there, please. Go
Can
ahead.
systems biology be used to understand how an environmental exposure affects an organ like air pollution in lungs?
Absolutely. Eventually, I am certain that those kinds of problems will be problems that are actually, I think, even now can be addressed by systems biology. One might imagine using these, for example, microarray-based approaches even, to be able to start to probe questions like you raised. Another strategy that's being employed is to use model organisms exposed to those same compounds to try to understand at a cellular level what might be going on, and then to relate it to the whole tissue, the organ, et cetera, et cetera. So absolutely. Oh yes, which reminds me, I failed to answer the second part of the other question about the influence of environment and on interplay between genes and the environment. That is an incredibly interesting thing about which we know, I think, very little about that. And it's, so I don't know at this point. Well, in some sense, that is a problem that is being addressed in systems biology, where people can take identical organisms and cells and look at differences that occur due to random fluctuations within the cells. So this isn't really interplay between genes and the environment, but is looking more at variation that's generated by inherent randomness in biochemical processes. People, of course, take identical cells and subject them to different environments, but I don't think those experiments really address the fundamentally interesting issues you raise. And it's just hard to deal with experimentally. Yeah.
Yes.
So over the past 2 decades, perhaps, NIH has funded systems biology, cell biology very well. And we have all this incredible information happening in the genome. But what was neglected over those years is the truly integrative nature of research. So, for instance, society I represent, we hear from some of our industry members that they cannot find people who can put the car back together.
Absolutely.
That truly understand that the systems are intact. That it's great to understand the working parts, but and it's easy to break the car apart, but nobody can figure out how to put it back together. So you seem to agree with me. I was just wondering if, as a solution, is there a proper balance where we need to start training people, because nobody's being trained in this type of effort?
Yeah. So I think your point is a very good one, and I would just comment that systems biology is a relatively, I would say, new thing in the past 5 years, and has become not even 10 years, 5+ years. And it's become, so part of the reason, so yes, we lack people who are appropriately trained. You're entirely correct. And but it takes there's some sort of lag time in changing training and et cetera, and changing funding for this kind of effort. And so NIH and NSF as well have started some very good programs to address the issues you point out. The lack of integration, both at the sort of on the research front per se, as well as on the training front. So on the research front, NIH has sponsored, actually the center I run has one of them, large center grants that bring together biologists and physical scientists to work on problems in systems biology using these integrative approaches. Some of the centers are focused more on physiology, some on more molecules like I've talked about. So there are some of those grants. It's perhaps not enough, and it's very hard to change the culture, and without the appropriately trained people, that's a tough thing.
But I was speaking more specifically perhaps to integrative organ.
Oh, sure.
So in intact animal, the best predictive model would be if you have a drug, put it in an animal or a human body, and people aren't being trained with that. So when I say integrative organ, integrative systems, I mean more integrative organ type training and research as opposed to bringing in
Sure.
physical science.
That's a whole other level of integration, sure. Yeah, I mean, but I would argue, I guess, that there are some training programs, et cetera, that are focused on that. That is more systems physiology kind of approaches, and even bordering on sort of pharmacology, what you would have called traditional pharmacology.
Dr. O'Shea, thank you very much for a splendid lecture.
And I'd like to say we would like to thank the Joint Steering Committee for Public Policy for co-sponsoring this luncheon. And also, I want you to know that immediately after your August recess, on September 13th, the subject will be chimeras. What are they and how they could be useful? I looked it up in a dictionary and said there's some kind of monsters.
Yeah.
But anyway, we look forward to your coming back, and please keep the NIH in mind. Thank you very much.